Hello, my name is Evan Kalb, and I'm going to try to get us back on track. I'm going to go through this really quickly and then pass it off to Andre, who I think is going to really give a little bit more detail to what we did here. But ultimately, what I'm interested in is tRNA. So my work looks at utilizing tRNAs to control and enhance cell-free translation systems. My thesis work kind of looks at overexpressing E. coli tRNAs, purifying them out, and building in pure, kind of detailed and discrete tRNA pools for enhanced cell-free translation system. And ultimately, that kind of lent a skill set that was useful for Andre's project, which is using JCVI Syn3A as a exciting minimal model of the central dogma. A reduced genome kind of offers an enticing look at what is just required for not only just a central dogma, but a self-replicating central dogma. So traditionally in cell-free translation systems, we use either pure, which is the as minimal as you can get, 36 purified proteins, ribosomes, energy mix required to drive those reactions, NTPs as substrates for transcription and translation, amino acids, of course, to build proteins. And these are all built upon E. coli elements. And then if you want to extrapolate to the most complicated version of E. coli within an in vitro environment, at least, is a lysate-based system where you crack open the E. coli and use its native cellular machinery to do virtually the exact same thing, but it's a little bit less unknown. We kind of think of this as a translational black box. There are, ancillary reactions that are going on that are important for increasing the yield. So what's interesting about JCVI Syn3A is it kind of toes this line between being closer to pure in terms of its simplicity, but still is a self-replicating central dogma. So one of the ways that we want to study that is to prepare a new cell lysate cell-free translation system based on Syn3A. And this is a ton of work. Andre worked really, really hard to get this up and running. It was a multi-year project, and his fortitude and patience paid off. He was able to build a new cell-free translation system based on this non-model organism. And what this requires is a lot of optimization, growth conditions, when do you harvest, how do you harvest, the buffer that you harvest in, and then subsequently the
Evan, we don't hear you suddenly. I don't hear you.
Looks like you're muted.
Evan, you're muted.
How about now?
How about now? Yes, much better.
Okay, perfect.
Repeat the last 30 seconds you said, please.
Okay, so long story short, there's a ton of optimization that is required in building a brand new non-model cell-free lysate system. And Andre did a ton of work, and a common optimization is actually just doping in E. coli isolated tRNAs. And when Andre did this actually didn't work. He got very little enhancement, or at least not very reproducible enhancement of translation. So what I did is I grew up M. capricolum, which is related obviously to the JCVI Syn3A, and just bulk extracted out the tRNA. And here it is on the gel, behaving as tRNA ought to at its length. And sure enough, that increased the translation in a reproducible fashion. So when I looked into a little bit more why this might be, it wasn't all that surprising. When you compare the percent identity matrix of the tRNAs of JCVI Syn3A and compare them to MCAP, they're nearly identical. They're roughly the same number. They're both are roughly 30 tRNA genes. Compare that to E. coli that has 46, and as a gamma proteobacter, has different identity elements and can become, not a very useful substrate for that kind of cell-free translation system. Another thing that's really intriguing to me about this system is Mycoplasma capricolum. And JCVI Syn3A kind of shed light on the importance of tRNA post-transcriptional modifications. tRNAs are very heavily modified. One of the things that I really like about this figure is that besides illustrating the shared post-transcriptional modifications between MICO and JCVI Syn3A, is you can start seeing just how important these modifications are. I want to draw to your attention really briefly tAA. tAA is a deaminase that turns adenosine into inosine, which is actually necessary for translation in much larger organisms. So this is a required gene in E. coli, but it appears to be dispensable in Syn3A, right? Because it's not present in the minimal cells. So it's interesting to think about what's going on genomically there. That inosine is supposed to allow for greater translation of a swath of codons through wobble decoding. So this is kind of a fascinating thing that I would be interested in looking at more. But something that I'm really interested is this whole concept of doping in E. coli MRE600 tRNA as a common optimization step seems kind of inappropriate to some of the new cell-free translation systems that are coming out based on non-model organisms. So for example, Vibrio natriagens or Vmax is garnering a lot of attention because of its incredibly fast doubling time of less than 10 minutes. Its genome has annotated 103 tRNA genes relative to E. coli's 46. To me, that seems like an obvious application of this technique to see if we can't bolster those translation yields. Streptomyces venezuelae is another interesting look. It's GC rich, and that is going to intrinsically have a different codon optimization than E. coli, but of course, they still doped in E. coli MRE tRNA. And then Bacillus subtilis is another example where tRNA has been doped in with varying levels of success. So ultimately, I think this project kind of illustrates the importance of utilizing native or at least near native tRNA in your cell-free translation systems. And I'm interested in pushing this further towards new and exciting platforms like Vibrio and the like. But with that, I'll take questions if there is time.
There is, Evan. So I think about, compared to E. coli cell-free systems, everything else is terrible.
It's garbage.
And is this approach of using, instead of using the native or almost native tRNA to supplement the cell-free system, is this a common strategy in cell-free systems, or is this something new to you?
I think it's new to us. I've been kind of digging more and more into the literature, and I haven't seen a ton of people working with this. The closest thing that I got to was one of the first groups to make Vibrio natriagens into a cell-free system, kind of utilized an equivalent of the P-rare plasmid, where they were encoding Vibrio tRNAs on a plasmid and co-expressing them, much like you would with the Rosetta strain from E. coli to kind of add more of the rare tRNAs to that system. But that's as close as I've seen in bolstering the amount of tRNA in these cell-free translation systems, not just wholesale bulk tRNA extraction and doping into the cell-free translation system.
And then the other question is, the gene that catalyzed the conversion to inosine, is that in capricolum and wild-type Mycoplasma mycoides, or is this something that the mycoplasmas have just done away with?
So it is in Mycoplasma capricolum. I don't know about mycoides.
It probably is then.
Yeah.
Okay. Other questions for Evan? Okay. Well, thank you. Nice job.
Hey, John.
Yes,
hey, John,
Kate.
There's a question in chat.
Oh. I can even possibly read this. Could you make all Mycoplasma tRNA in coli with the right post-transcriptional modifications?
Interesting. That would free up a lot of work, right? Because it took a little while to get this bug to grow. Capricolum is, if we would have just done straight Syn3A, it would have taken a much longer time. The only issue that I would be skeptical about doing is getting non-native modifications on that tRNA, right? So E. coli, sure, they have shared post-transcriptional modifications, but there are some that are going to be overly abundant. Let me see if I can't pull up my slide here really quickly. For example, if you take a look, there's not a thymine in the T-size stem loop of Mycoplasma capricolum. Usually what we see is a thymine right next to a pseudouridine in that loop. So that could be an example of adding in post-transcriptional modifications that aren't related to the machinery. Now that might have its own interest, like maybe those would enhance cell-free translation more by getting those non-native modifications. But to answer your question shortly, yes, it would be interesting. I'm not sure how much of an impact it would make.
As a result of minimization, did we eliminate some of the genes that, or the proteins that modify tRNAs? Do you know?
Yes. So, or at least you got rid of tAA and this YBK. And these are particularly interesting ones because, like I said, tAA is indispensable in E. coli and higher prokaryotes. And then this YBK encodes for cytosine methylation and uridine methylation. These are important modifications for maintaining the open reading frame of a tRNA. So it's pretty fascinating that you got rid of these. I'm especially surprised about tAA because of its ubiquity in prokaryotes.
So it would be relatively straightforward to put these genes back in and see if it affected growth rate.
Yes, and I would be really interested to see if it actually did enhance growth rate. This is something that I was thinking about with Andre, where by putting in Mycoplasma capricolum tRNA into this SYN3A system, we're probably putting in a better, it's not often that you put in a better tRNA pool than the native tRNA pool. And I think this is an example of it, right?
Well, so, this is an experiment that we could work with you on. You can chat with us about this, another time, but we could do these changes or teach you how to do these changes and the whole experiment. Thank you, Evan. So next up, my buddy Andre Sake from Radboud University, who
John?
The most patient man I know. Another question?
Yeah, I had yes. Hi, John. I had a comment for Evan. As you probably know, John, we published a while ago a study looking at translation genes that are common, that we found in different Mycoplasma species. And if you look in one of the branch of the Mycoplasma that we call the hominis branch, that is very large. In fact, the tAA gene is missing in all of them, in fact. And so so John has built something that was already done in other Mycoplasma, in fact. But yeah.
That's very interesting. Thanks, Alain.
Is it in the pneumoniae group?
I have to look again, but I think so. I have to look again. I have the figure under, but I will send the reference in the discussion for the paper, and I think I see it in the pneumoniae group. I have to verify, but at least it's missing. It's missing in the hominis group. Yeah.
I've sequenced so many of these, I should know this, but
yeah, you should know that.